So first, good afternoon or good evening, dear colleagues and dear members of the organizing committee. I would like to thank you for inviting us to present our data and for excellent organization of the conference. Today, I would like to talk about comparative analysis of cell-based product obtained with different systems for isolation of stromal vascular fraction. And this study was performed by the National Medical Research Center for Rehabilitation and Balneology in Moscow in collaboration with scientists from Gamaleya National Research Center and Moscow Clinical Scientific Center. Oops, it doesn't work. I will try this. It's very slow. Okay. As you probably know, the use of subcutaneous PET as a source of progenitor and stem cells for regenerative medicine has become widespread in the last decade. And the main advantage of using adipose tissue is as a source of stem cells when compared to bone marrow is less invasiveness of the sampling procedure during liposuction and significantly larger number of mesenchymal stromal cells obtained per 1 unit of volume of the tissue. And currently, a stromal vascular fraction is widely used as a highly effective source of cells based on human material that can be obtained with minimal intervention. The stromal vascular fraction contains multiple cell types like adipose-derived stem cells. Sorry. Sorry, I'll try to explain it. Okay, it will be better. Mesenchymal or endothelial progenitor cell leukocytes and pericytes and vascular smooth muscle cells. Okay, and in this case, the development and use of special devices for stromal vascular fraction isolation from adipose tissue is very important for widespread use of stem cells in medical practice. In addition to obtaining cell products using enzymes technologies, new technologies based on mechanical isolation procedure has also been applied. However, the use of different methods of adipose tissue isolation can lead to a variability in can lead to variable yields of cells in the final product. The aim of our study was to perform a comparative evaluation of cell product obtained from human adipose tissue using two different systems based on enzymatic and mechanical isolation procedure. The adipose tissue samples we obtained from 14 healthy females who underwent liposuction surgery at the Clinic of Plastic Surgery. All patients provided informed consent before surgery. And the clinic established standard inclusion and exclusion criteria for liposuction. For example, criteria for inclusion were the presence of local fat deposits in the abdominal area, lateral areas of the waist, back, and knees, with the skin fold thickness in the investigated areas not less than 2.5 centimeters, but no more than 6 centimeters. And criteria for exclusion were as follows. It was obesity or too dense structure of adipose tissue, impaired skin integrity, heart disease, bleeding disorders, diabetes, malignant neoplasms, and acute infection diseases, mental disorders, and some other. Materials and methods. We used two systems. First system for enzymatic isolation. It's a SWIFT system was recently developed by a joint company, and the other system, so-called double syringe system, was used for mechanical isolation of stromal vascular fraction from adipose tissue. First, I will talk about this system, which was recently developed by joint cell company. This system was used for enzymatic isolation of stromal vascular fraction. This system uses a disposable separator, which is a transparent plastic container with two chambers, and the system also includes an enzyme collagenase. Some words about this separator. This separator has two chambers, the upper chamber, and the lower chamber, separated by a nylon filter with pores of 100 micrometers. These chambers are separated by a cell filter, and the system also have a special bacterial filter on the cover, and there are 6 channels for the introduction of components. Adipose tissue can be introduced through 4 channels, and 1 channel is used for removing supernatant, and 1 channel is used for removing stromal vascular fraction from the lower chamber. The isolation of stromal vascular fraction was performed according to the approved protocol, and the procedure included several steps. First step is adding lipoaspirate up to 200 milliliters to the separator, then washing the lipoaspirate with Hartmann solution, and then it was followed by the addition of the collagenase and incubation in a shaker. Then this separated with the content was followed by centrifugation of the separator. It's 300 G for 10 minutes, and then aspiration of supernatant, and then washing with the buffer. And then we can withdraw the stromal vascular fraction from the lower chamber, and after sampling this obtained cell fraction were studied using microscopy, flow cytometry, and cell culture methods. Another system that we used, it's originally it was designed to obtain platelet-enriched plasma, and now due to this, the use of mechanical methods, had become widespread. This system has been adapted to the mechanical isolation of stromal vascular fraction. So this double syringe system uses the largest syringe with a volume of 15 milliliters and is used to collect lipoaspirate, and the smallest syringe is used to withdraw fraction from the larger after centrifugation. And also there are 2 adapters which can be used to adjust this double syringe to a normal syringe with mesh diameters of 2.4 and 1.4 micrometers, and they are used to disperse this adipose tissue to obtain cell fraction. The maximum volume of fat tissue that can be processed with this syringe is 15 milliliters, and we use Anatrix double syringe system in our study. To compare the efficiency of these two systems, the obtained cell products were evaluated to several parameters. First, it's like the volume of residual oil and cell phenotype and total number of nucleated cells, viability of nucleated cells, and some others. First, using flow cytometry, we showed that the isolated cell products were characterized by the presence of endothelial progenitor cells, vascular endothelium, pericytes, and adipose-derived stem cells. But it should be noted that percentage of adipose-derived stem cells in the cell fractions obtained from using the system with mechanical isolation was very low. Then we compared the parameters obtained with these two systems, and the result showed that the method of adipose tissue processing has a significant impact on the characteristics of the obtained cell products. For example, we can see that the number of nucleated cells and the number of nucleated cells per unit of lipoaspirate, as well as viability of nucleated cells, were significantly higher in the cell product obtained with the enzymatic system. Also, we performed a microscopic examination, and it showed a significant difference in the number of isolated cells per unit of adipose tissue and per unit of lipoaspirate and cell viability when we're using the enzymatic and mechanical systems. Compared with the enzymatic method, the mechanical system produced product with higher volume of residual oil, number of destroyed cells, and connected tissue particles. And this our study is still in progress, and we can see that it's very important that how adipose tissue is processed by a mechanical or enzymatic way has a significant impact on the characteristics of the stromal vascular fraction obtained. When isolated mechanically, the obtained cell product is characterized by a low yield of nucleated cells, large amount of residual oil, and damaged connective tissue in the final product. In the enzymatic isolation, the cell product is characterized by a high yield of nucleated cells, which is very similar to the number obtained with the manual enzymatic method. And also, this fraction is characterized by high proliferative potential of adipose-derived stem cells. Thank you very much for your attention.